This is Critical Thinking on the Digestive Physiology Lab, and here are the questions. So this is the first one. Second one, this is on carbohydrate digestion, and this one is on fat digestion. Let's look at the first one, which is more or less a general question which asks you some of the important factors that need to be present when you perform activities on digestive enzymes. And this is obviously outside the body. So let's look at these and see which one is not required. So the first one is maintenance of proper temperature. So this is important because if you want to look at digestive enzyme activity and especially want to see how it will resemble the activity in the body, you need to maintain proper temperature, and you would like to maintain it at the right body temperature, which is around 37 degrees centigrade. So this is extremely important because if the temperature falls, then obviously enzymes will not be able to act properly. Or if the temperature is too much, for example, if it gets very high, like if you boil the substance, then enzymes get denatured. So, they can't act then too. So maintenance of proper temperature is very important. The second one is control tubes. You always need control tubes when you do activities of this nature because you want to make sure that there is no contamination. That means that all the ingredients that you are using, they are pure. So that's why you run certain tests in those control tubes where you use just the ingredients without the enzymes and see whether you get any activity or not. And if the substance is pure, then based on what kind of test you're running, it should sort of show up accordingly. I mean, you should see that there is no contamination. So control tubes are very important. You have to maintain a proper pH because that's how you will be able to mimic what happens in the body. So for example, certain enzymes act at a neutral to slightly alkaline pH, certain act at an acidic pH. So you have to maintain a proper pH in order to see optimal enzyme activity. So that's also very important. The fourth one is means to conduct enzyme assays, or in other words, you have to have certain means to see what are the end products of digestion, be it by means of reagents where the color changes, or you put it in to a photometer where you look at the optical density. So you have to have some means to find out the end product of the digestion, and that's known as enzyme assay. So that is important. The last one, which says presence of hormones. Now you don't need this in order to carry out activities on digestive enzymes outside the body when you're doing an experiment because the enzyme is already there. Remember, in the body, hormones bring about the release of enzymes. So in the body, your hormones are needed because they act to cause secretion of enzymes from the cells. But here, you're already working with enzymes, so you don't need the hormones. So hormones are not needed. Hence, this is the answer to this question. Now let's look at the next one, which talks about carbohydrate digestion. And it says that it begins in the mouth because of the presence, obviously, of salivary amylase. So from the salivary glands, you have salivary amylase, which is produced, and that acts on carbohydrates. However, when the food enters the stomach, this carbohydrate digestion stops. And the reason is, let's look at the choices. It's because pepsin is present in the stomach. No, this is not a choice. Pepsin does not interfere with digestion of carbohydrates by salivary amylase. Pepsin acts on proteins, but it does not interfere with salivary amylase. In fact, in the fundus of the stomach, especially if you eat a large meal, where there's some food in the stomach, in the fundus of the stomach, carbohydrate digestion can still continue because of the amylase, which is probably coating the food bolus. And that's because when you eat large meals, the fundus does not move much. So the food over there, it's not churned too much. So the amylase continues to act. But once that food comes down into the rest of the stomach, the amylase will stop acting. And it's not because of pepsin. Pepsin does not interfere with it. Neither does intrinsic factor. Intrinsic factor is only responsible for absorption of vitamin B12. And it does not interfere with salivary amylase either. So that's not true. The third choice, which is pH is too low. this is the correct answer, because in the stomach the pH is acidic, whereas in the mouth the pH is neutral to very near neutral. It's about 6.7 to around 7. So this is, you can see the pH is much higher than it is in the stomach. And pH is very important for all of the enzyme activities, as we saw in the previous question. Now, because the pH is too acidic, therefore amylase cannot act. It kind of gets deactivated. It's not able to act in this low pH, and hence carbohydrate digestion stops in the stomach. So this is the answer to this question. And obviously this last one, which says no salivary glands are present in the stomach, that's not the reason why carbohydrate digestion cannot occur in the stomach, obviously. So the correct answer is because the pH is too low. Now let's look at this last question, which asks you which of the following statements regarding fat digestion is incorrect. So the first one is fat digestion occurs in the small intestine, and this is a true statement because in the small intestine you have the presence of lipases, which are the pancreatic lipases. So they make their way into the duodenum through the pancreatic duct, and the pancreas is the only significant source of lipases. You have a little lipase in the mouth, but that really does not cause any digestion. So for all practical purposes, fat digestion begins and ends in the small intestine. So this is true. The second is bile salts and lipase are needed for proper fat digestion, and this is also true. Bile salts are needed because they break the fat down into smaller particles, allowing the lipase to act on them and then break the fat into fatty acids and glycerol. If bile salts are not present, you will find that fat digestion is improper. So in order for lipase to act, the presence of bile salts is very, very important. So what they do is they emulsify, or they break down the fat into emulsify. They break down the fat into smaller particles, allowing the lipases to act on them. So this is true also. And chemical digestion of fats produces fatty acids and glycerol. As I mentioned earlier, the lipases act on fats, and so triglycerides are broken down into fatty acids and glycerol. So fatty acids and glycerol, and that is the end product. And lastly, the pH should be acidic. Now this is false because in the small intestine, the pH is neutral, very similar to what it is like in the mouth. So the pH should not be acidic. In fact, when the acidic chyme comes into the small intestine, the pancreas produce a bicarbonate-rich pancreatic juice. So the bicarbonate buffers the acid and neutralizes it, as also the intestinal cells produce mucus in order to neutralize the acid and bring the pH, raise the pH and make it closer to neutral. So the pH should not be acidic. Instead, it should be neutral. So this is the incorrect statement, and hence the answer to this question.